Polymerase chain reaction, PCR. Since the emergence of COVID-19, more and more people have heard about PCR. It is now the most common diagnosis for this viral disease. But what is a PCR? PCR is the acronym of Polymerase Chain Reaction. It is a chain reaction catalyzed by a DNA polymerase. DNA polymerase is naturally present in most of the living organisms. From the name polymerase itself, you can already guess this enzyme's function is to make long chain of substance. And chain reaction means make more copies of this polymerase substance. And what is being polymerized? The answer is the nucleotides, also known as bases. There are 4 of them, namely adenine, thymine, cytosine, and guanine. The DNA polymerase extending the chain by complementing the DNA template provided in a PCR. A is always paired up with T, while C is always with G. A typical PCR can be set up by mixing together 6 major components. First, Taq polymerase, which is a thermostable DNA polymerase derived from extremophile Thermus aquaticus. The magnesium ions is an important cofactor for Taq in DNA polymerization. Next, PCR buffer provides the Taq with the optimal pH for its activity. And it also contains potassium ions which help promote the primer's annealing. A mixture of 4 nucleotides is essential for the extension of target DNA chain led by a pair of custom forward and reverse primers. Finally, the DNA template which the Taq can copy and extending its nucleotide sequence. But these components alone are still not enough to initiate the chain reaction. Taq polymerase activates and extends the DNA at a temperature of 72 degrees Celsius, but double-stranded DNA needs more than 95 degrees Celsius to be separated. Whereas primers, which actually are very short pieces of DNA, can only anneal to the separated template DNA strand at a temperature ranging between 45 to 60 degrees Celsius. So, to initiate the chain reaction, the mixture needs to be subjected to 3 different temperatures successively. First, 95 degrees Celsius is to separate the DNA double strands, and then followed by 45 to 60 degrees Celsius. The exact temperature depends on the primer sequence. This is for the primers to anneal. And finally, 72 degrees Celsius is for Taq to extend the target DNA sequence. In order to have the chain reaction continuous, thus generate more copies of target DNA, the 3 temperatures need to be looped. An initial denaturation step at 95 degrees Celsius is first needed to separate the double-stranded template DNA. In each subsequent cycle, 3 temperatures are applied successively. Then the loop continues. The DNA copies are then increased exponentially and accumulate until the end of the PCR. This exponential growth characteristic of PCR is extremely useful for detecting traces amount of DNA. By the end of the 35th cycle, as many as 68.7 million copies of DNA is yielded, with a mere 2 copies of DNA at the start of a PCR. The exponential growth of DNA copies will eventually be dwindled by the exhaustion of nucleotides, primers, or the loss of Taq activity due to prolonged incubation. The result of PCR is resolved using a common technique known as gel electrophoresis, then visualized with a specially designed imaging system. The intensity of the white color bands correlates with the amount of accumulated DNAs at the end of PCR. One important variation of PCR, known as quantitative PCR, is now widely used for COVID-19 diagnosis. Note that the causal agent for COVID-19, SARS-CoV-2, is an RNA virus, and PCR can only be applied to DNA. Hence, a preceded reverse transcription step is needed. The viral load can then be detected and quantified by means of qPCR through the observation of CT or threshold cycle. In the next video, I will bring you into the lab and show you how PCR is done.